go ahead and get started. Let's see, before we start with the actual material, let me, remind you of what's coming up in the next 1 week and 1 day. So we have class today, we have class on Wednesday. That's going to be our last class, Wednesday. Then there is your lab practical quiz on Thursday or Friday. I sent out an email and I posted on Moodle the schedule for the practical quiz administration. So make sure you check the time and arrive on time for your quiz. You don't have to arrive, arrive a few minutes before your quiz time. You don't have to arrive at your ordinary time. It's just wait around for it if your time is after the start time of the lab. That's the practical quiz. And then a week from tomorrow is the final exam. The schedule is a little funny in the sense that the time allocated for this final exam isn't really enough. So I've broadened it, which means I'm stepping on a time allocation for another course. And I asked if you have another final exam scheduled for Tuesday, let me know, and we'll work out an arrangement. I haven't heard from anybody that I've got 2 final exams scheduled back to back, 8 to 10 and then 10 to 12. I haven't heard from that, but if that's what your schedule says you're doing, let me know and we can make arrangements. If I don't hear from you, I'm going to assume that the start time of 9 a.m. for the A and C final exam on Tuesday, a week from tomorrow, is okay. I'm trying to think if there's any other announcements or course administration things. There is, you may have already received an email about course evals. I think it went out today, or it went out before, but I think today is when the evaluations actually open. So, as always, I welcome your feedback on the course evals. It's a little strange this year because the evaluation won't affect my future courses at Earlham anyway, but it might affect my future behavior, so I welcome your evaluation. It's, it's online and it's now anonymous. You're not asked to sign it anymore. I think that covers it. If I think of something else, I'll let you know, or we can talk about it on Wednesday. So here's where we've been and here's where we're going. We're going to reproductive systems, starting with male, and then we'll take a break, and then we'll end with female reproductive anatomy and endocrinology. Just a note, we're talking strictly biological male and female reproductive anatomy and physiology. We're not talking about gender. We're not talking about other social constructs surrounding male and female male behavior, female behavior. That's not, I'll mention some, stereotypical male behavior, and, we'll talk about female-ish female behavior, but mostly we're focusing on reproductive anatomy and physiology. Online people, are you hearing me okay? Yeah. Okay, good. I'm not seeing the little lights on the machine the way I usually do. Okay. Are you still hearing me okay? Yeah. Okay. So just to be clear, we're talking about biological sex. We're not talking about gender. So here is an anterior view of the male reproductive organs. The anterior view isn't very informative. Here is a view from your textbook, midline sagittal section of the male anatomy. Let's see. So just to get oriented, anterior, posterior, inferior, superior, right? And of course, this thing here is supposed to cartoon the colon, the descending and the sigmoid colon, which is not part of the reproductive system, but that just gets you oriented. Posterior. All of the other names on this slide are anatomy that I could test you on the final exam. Oh, the final exam, just to be clear, is going to be at least 2/3 and maybe more section 3, and then some cumulative. Okay, so the final exam is partially cumulative, but mostly endocrine and reproductive anatomy and physiology. So the anatomy on this slide could be quizzed or tested on the final exam. So just to be clear, the penis has several structures within it that I am going to ask you to learn the name of. Interior structures include the corpus cavernosum and the corpus spongiosum that are vascular tissues associated with erection. The male penis has a foreskin, which has a Latin name, prepuce. I'm not sure I'm saying that right. The head of the penis is called the glans. The tube that is going to deliver sperm and semen is the urethra. It's the same tube that delivers urine from the urinary bladder. But that's not what we're talking about in this setting. The scrotum contains the testes and the epididymis. We'll talk about those structures in just a few seconds. There's a tube from the epididymis, wraps around the urinary bladder. This is the tube for delivering sperm to the prostate gland and the seminal vesicles, where other material is mixed to form semen. This one is labeled, yeah, prostate gland. And then during ejaculation, semen, including sperm, the male reproductive cells, are delivered. So, there is a muscle that I do want you to know the name of. It's a muscle that is innervated by the sympathetic nervous system, and it is important for ejaculation. We'll talk about erection and ejaculation in a few minutes. So there's the anatomy, the gross anatomy, the whole body anatomy that I do want you to know for the male reproductive anatomy. This is a blow-up of the testes. Just to be clear, testis, singular, testes is plural. Okay, so here's one testis. Again, some, not microscopic, but smaller level anatomy that I do want you to know. So the surface of the testes, this is not there's skin on the outside here. So the scrotum is the name of the sac, the skin and fat and other tissues that surround the testes. The testes has a membrane on the outside called the tunica albuginea. And I'm going to ask you to know that name because it's actually a name that is used in the female as well. So that's a name to know. Female anatomy. The testes is formed of lobules separated by septa. So each of these brown cartooned areas is a lobule, and the membrane in between them is called a septum. One septum, several septa. Inside the lobules are tubules that are important, the name of which is anatomy, seminiferous tubules. So these are all seminiferous tubules within the lobules of the testes. The seminiferous tubules merge into this network called the rete testis. Rete, I think, just is Latin for mesh. Then this meshwork of tubes enters the epididymis. There's sort of a complicated set of tubes in the epididymis. The tubes are now called efferent ductules. So just the name of the tubes, seminiferous tubules, rete testis, efferent ductules that merge to form the vas deferens. The tube that then connects the testes wraps around the urinary bladder to the prostate gland and then to the urethra. Okay, let's get a little bit more, let's get more microscopic. So this is within the testes. This is supposed to be a cartoon of a cross-section of one of the seminiferous tubules. So we're zooming in on a seminiferous tubule and looking at it in cross-section. The seminiferous tubules are mostly lined with a cell type that I do want you to know the name of, called the Sertoli cell. The Sertoli cell lines the seminiferous tubule. It is going to respond to a hormone that we're going to get to in a minute called FSH, follicle stimulating hormone. It's going to respond to FSH and support spermatogenesis. So the development of the male gamete, sperm, the sperm is a separate cell. The sperm does not come from Sertoli cells, but Sertoli cells provide support and are actually required for spermatogenesis, the development of the sperm, from their own cells. So, okay, we'll talk about spermatogenesis in a minute. In addition to the Sertoli cells that make up the seminiferous tubules, there are interstitial cells, cells that are outside of the tubule. So some books just call them interstitial cells. I call them, and this book also calls them Leydig cells. Either name is acceptable, but I like Leydig cells because the L tells you that they respond to LH. So Leydig cells respond to LH, a hormone that we're going to get to in a minute, and produce testosterone, produce the androgens that are important for male reproductive anatomy and physiology.
Bob,
Yes.
are there any analogs to Leydig cells elsewhere in the body, or are the testes like the main production center for testosterone?
There are no analogs for Leydig cells anywhere in the body, but the testes are not the only source of androgen. It turns out that the adrenal cortex can make small but non-trivial amounts of androgen, including testosterone.
Thanks, Bob.
Okay, so now let's talk about spermatogenesis. That's cartooned in this slide, or this part of the slide, the development from precursor stem cells, diploid cells, using the terminology of CGI. We're going to generate, from spermatogonia, we're going to generate spermatids, sperm cells. This slide doesn't go through the details, but this slide does. So the precursor cell, and the reason I'm doing this in the male is because we're going to do something very similar in the female. The precursor cell for sperm are spermatogonia. I do want you to know that name. Those are, diploid human cells. They're not yet haploid gametes. Now there's something weird about this slide that I need you to realize and internalize. This slide, and I need you to do it now for the male because we're going to have the same problem when we talk about female oogenesis, egg development. The problem is that this slide cartoons the development as if it is moving in space laterally. It's not. The cells are moving in space vertically through these spaces in between the Sertoli cells. So this is the outside of the seminiferous tubule. This is the inside of the seminiferous tubule. These are layers, stacks of Sertoli cells with spaces in between them and some junctions. The spermatogonia develop and migrate through this space. So this spermatogonium is going to develop according to this cartoon pathway, but it's not going to migrate laterally, it's going to migrate vertically. The reason they put it horizontally is to spread it out so that you can see it. So this point right here is illustrated over here, but it's happening to this spermatogonium here. Okay? So don't get confused by the idea that there's lateral movement. It's not a big deal. It's just that these slides are confusing if you're thinking about, wow, how did this cell get all the way over here? It's not getting all the way over here. It's getting all the way over here. Okay? Anyway, in development, spermatogonia become primary spermatocytes, which are still diploid. They migrate through this junction. Let's not worry about the tight junction. They migrate through the tight junction and then become secondary spermatocytes. This step is meiosis 1. I'm not going to go back and review CGI level mitosis and meiosis, but just so you know, this step is meiosis 1, and this step is meiosis 2. These cells are the haploid cells, right? Remember, after meiosis is the generation of haploid germ cells, or gametes. That's what's happening here. So a spermatocyte becomes a primary spermatocyte, becomes a secondary spermatocyte, become spermatids during meiosis. We get 4 spermatids per primary spermatocyte. These spermatids then continue to migrate and develop in the spaces here and become mature spermatids. The spermatids also has another name, spermatozoa, or just sperm cells. Okay? But sperm cells is a little vague because all of these are sperm cells just in various levels of development. But if you call this a mature sperm cell, I will accept it. But there is a jargon here, of course. Spermatogonia, primary and secondary spermatocytes, immature and mature spermatids, or spermatozoa. Just the names of the cells. So in order for us to communicate at a high level about male reproductive anatomy and physiology, here are some more names to know. I have a few slides that show this cartoon. Well, here's a cartoon that shows it a little bit better in the sense that it's not migrating laterally, it's migrating vertically. So spermatogonia, primary and secondary spermatocytes, immature and mature spermatids. So this slide is a better cartoon of the way this actually works. Around the cells are the Sertoli cells that support and are required for this development. Here is a stained cross-section of seminiferous tubule, Sertoli cells. Somewhere in between migrating are the spermatocytes and the spermatogonia, spermatocytes, spermatids. These fuzzy things are the tails of the sperm cells, the tails of the mature spermatids in the lumen of the seminiferous tubule. Out here are the interstitial Leydig cells that produce testosterone. Another slide available on Moodle. I didn't print it out for you, just showing you that the cartoons are somewhat realistic in terms of the development in terms of the way it looks, especially cartoons like this. Electron micrograph, again, just more evidence for that the story I've told you about the development is real. These are the tails of the mature, or flagella, of the mature spermatids. Okay, now let's transition to endocrinology, because the endocrine system controls this process. So let's start from the top and work our way down. So we're going to start at the top in the brain, and then we're going to work our way down into the gonad, into the testes. And we're going to do that with the female reproductive endocrinology as well. Male is simpler, but the themes in the male reproductive endocrinology story are going to be useful for understanding the more complicated female reproductive endocrinology story, because some of the players are the same. So, the hypothalamus responding to various stimuli, diurnal rhythm, other input stressors or non-stressors, releases GnRH. So gonadotropin releasing hormone is released from the hypothalamic neurosecretory cells into the median eminence, where it flows through the blood, through the portal circulation to the, there it is, the portal circulation to the anterior pituitary. The cell type we're focusing on here, in both male and female, are the gonadotropes. Gonadotropes are the cells that express GnRH receptors, and in response to that, synthesize and secrete two hormones that are actually structurally related to each other: FSH, follicle stimulating hormone, and luteinizing hormone. These names, follicle stimulating hormone and luteinizing hormone, those are names from female reproduction, but it's the same hormone in the male, and thankfully they kept the same name. Follicle stimulating hormone, luteinizing hormone are released from the gonadotropes of the anterior pituitary. Luteinizing hormone stimulates the Leydig cells to make testosterone. So there's a simple linear feed-forward story. Luteinizing hormone from the anterior pituitary, systemic circulation, gets to the testes, interacts with LH receptors on the Leydig cells, and tells them to make testosterone. FSH from the anterior pituitary, systemic circulation, interacts with, binds to FSH receptors on the Sertoli cells of the seminiferous tubule, and tells them a complicated set of processes to stimulate the development of sperm, spermatogenesis. We're not going to get in, this is complicated cell biology that we're not going to get into. But I do want you to know that the Sertoli cells are required to stimulate spermatogenesis. The testosterone produced by the Leydig cells, which are right around the seminiferous tubules, diffuse locally. We do not, this is paracrine. This is paracrine because it does not require a bloodstream. The interstitial cells, the Leydig cells, produce testosterone, and it diffuses locally to affect spermatogenesis of the nearby seminiferous tubule. Paracrine. The testosterone produced by the Leydig cells exerts negative feedback on the anterior pituitary and on the hypothalamus to keep this in balance and in control. In addition, Sertoli cells produce a protein called inhibin, which inhibits the anterior pituitary. So we have two forms of negative feedback. Testosterone exerting feedback on the anterior pituitary and on the hypothalamus. Inhibin exerting negative feedback on the anterior pituitary. Do not worry about the fact. So your book makes a point that inhibin is inhibiting FSH production and testosterone is inhibiting LH production. That's a detail I don't think you need to know. So you can cross these words out just to simplify your slide. But I do want you to know the feed-forward pathway and both feedback pathways. Because that's endocrinology, that's the reaching homeostasis. Positive feed-forward, negative feedback. We've seen it several times already. Testosterone does help the Sertoli cells help spermatogenesis, but of course testosterone has other effects that we'll talk about in a minute. Testosterone and inhibin are getting into the systemic circulation and going everywhere, and testosterone has effects other than in the testes only. Any questions about this, the feed-forward feedback story that I do want you to know? I asked you to tell me a story like this for the cortisol, in the cortisol story of the problem set. All right, let's start to talk about the details of the hormones. So now we're talking about male reproductive endocrinology. We are going to talk about LH, FSH, and testosterone. Let's start. LH is a protein. It's got that's all I want you to know. For your additional knowledge, it has two subunits, and one of the subunits is the same for FSH. I will not test you on this. I do want you to know that LH is a protein. It's a peptide hormone. What cells are producing it? The gonadotropes of the anterior pituitary. I do want you to know that. The stimulus for the release of LH is GnRH from the hypothalamus into the portal circulation. The target of LH are the Leydig cells, the interstitial cells in the testes. LH stimulates Leydig cells to produce testosterone. The receptor turns out to be a GS coupled receptor, but again, I am not asking you to memorize the nature of the GS, of the receptor. But I think I could test you in this cumulative part of the final exam. If I told you that LH, the LH receptor was a GPCR coupled to GS, and I asked you to tell me the story of LH, and I said, tell me everything you got, I would expect you to tell me the GS story. Okay? LH. FSH is very similar. FSH is produced by gonadotropes. It is a glycoprotein produced by gonadotropes. Its stimulus is GnRH from the hypothalamus going into the portal circulation and interacting with FSH receptors. The FSH receptor is also GS linked. The FSH, so I'm not telling you what the GnRH receptor is, but I could. If I told you the GnRH receptor, in fact, I can't remember. I think the GnRH receptor is also GS linked. Hint, on the final exam, if somewhere in the final exam you tell me the GS story, and then somewhere else on the final exam, I'm asking you, give me everything you got, and it's the GS story again, you don't have to tell me the GS story again. You just have to say, this is the GS story, see question number da-da-da, and I know you know it. Okay, so if you've written something out in great detail somewhere on the exam, and you are now realizing, oh, this is the same story as I wrote back there, just refer to somewhere else on the exam, save the few minutes that it would take to write out the details, but tell me you know that the gory details that I've asked you for in this story have a piece from another story. It's just a time-saving device that shows me that you know the stuff, but you don't have to write it all out again. Okay, so getting back to the FSH receptor is a G protein coupled receptor, and it stimulates spermatogenesis by the Sertoli cells. It tells the Sertoli cells, do what you need to do to help the spermatogonia develop into primary and secondary spermatocytes, help them develop into spermatids. Okay, now let's turn our attention to testosterone. This is a slide you've seen before. This is a slide I will not ask you to know, but I will ask you to be able to interpret like you did on the problem set. Testosterone and other androgens, so androstenedione is another androgen that is not the same as testosterone, but is similar to it and has androgenic activity. Same with DHEA, dihydro something androgen. All three of these androgens come from precursors that all come from cholesterol. So on the final exam, I will not ask you to be able to know this, but I might give you this table and ask you to solve a problem. It's kind of like I did on the problem set. On the problem set, I didn't give it to you, but you could look it up. On the exam, I would give it to you, and you would solve the problem like you did on the problem set. Knowing that P450 17-alpha hydroxylase is required here and here to develop the androgens, but is not required to develop, to make aldosterone, but is required to make cortisol, is something I could challenge you with on a question on the exam, if I give you this flowchart. Anyway, back to the same story. Androgens are produced from precursors, progesterone and pregnenolone, which come from precursors which come from cholesterol. Testosterone, the male steroid hormone. So now we're not talking about a peptide hormone. You know, we're talking about a steroid hormone. Produced by and released from Leydig cells. The stimulus is LH from the anterior pituitary. The target cells in the testes are the Sertoli cells to help them do spermatogenesis along with FSH. But there are many receptors for testosterone. Muscle, bone, brain, neurons. So here are some pictures of, male anatomy and physiology. A male characteristic of facial hair. I don't even remember what male characteristic I'm trying to illustrate here. Muscle. I guess I could be trying to also illustrate display behavior. Display behavior, boxing is not aggression, but testosterone is also associated with the male sort of aggression behavior. Risk-taking is associated with maleness. I'm starting to blur the boundaries between male sexual behavior and male gender behavior, right? So I'm on thin ice here, because obviously women do risky behavior, and women do display type behavior. And again, remember, I am not a believer in men and women, right? Gender is something much more complicated than sex. But there is some characteristic male phenotype associated with testosterone.
Bob.
Yeah.
So I actually don't know very much about this, but in terms of individuals who perhaps have an extra X chromosome, are their testes or testosterone production levels changed at all?
You mean an extra Y chromosome? Because a triple X is female, female, female. Remember, XX
I was
is...
thinking I was thinking like XXY. I'm not super sure about all of the recombination.
Yeah, I'm not sure of that either. An XXY genotype, I'd have to look up what the XXY genotype is associated with in terms of phenotype, reproductive anatomy, or sexual behavior.
Okay.
My guess is that an XXY genotype would be ambiguous genitalia, which we'll talk about in a minute. And I don't know where this person would be on the gender spectrum, or is likely to be on the gender spectrum. There's so many variables there.
Gotcha. Thanks, Bob.
So testosterone, a derivative of cholesterol. Again, I'm not going to ask you to know which one is testosterone and which one is cortisol, but you should be able to identify this four-ring structure as telling you it's a steroid hormone. So the development of maleness, of male genitalia, depends on the Y chromosome, the presence of the Y chromosome. The Y chromosome is a little stub of a chromosome. It has very few genes. But what it does do is it has one really important gene. So of all the chromosomes, the Y chromosome is the, the runt of the litter. It's this tiny little thing that doesn't do very much except create male phenotype. There's a gene. I am not going to test you on development. This is complicated. This will not be on the exam. But just so you know, the Y chromosome contains a gene called the SRY gene that produces a protein called the SRY protein. And this protein is essential to turn off the female pathway and turn on the male pathway. So the default pathway in humans, and probably in other, great apes, is female. Without SRY, we all develop as female. But some of us, with a Y chromosome and producing SRY, turns off the female pathway of development and turns on the male pathway, which then starts to produce the cells that make testosterone, androgens, and that continues, that sort of reinforces the development of male genitalia. The prostate gland, the seminiferous tubules, the testes, the penis, all of that requires SRY to overcome the default pathway, which is development as a female. In some species, like amphibians, there's a temperature dependence to this. So you may have heard that turtles, eggs at a certain temperature, all become female. And at another temperature, they're more likely to become male. It's kind of the same story. A default pathway at a certain temperature. Well, the point is in human anatomy and physiology, which is what you're interested in, not turtle physiology and anatomy. Human anatomy and physiology, it's not a temperature-dependent process, but it is a process that depends on this little runt of a chromosome, the Y chromosome. All right. So in order to talk about human reproductive behavior, we need to talk about sex again, or still. We're talking about the male erection. This requires the corpus cavernosum and the corpus spongiosum. So the male erection is a vascular thing. Well, let's just go through the story. So it starts with arousal. You know what I'm talking about. The parasympathetic nervous system is stimulated by the arousal and releases acetylcholine. In the endothelium of the blood vessels in the corpus cavernosum and corpus spongiosum. The acetylcholine there, interacting with receptors, stimulates the endothelium so that the acetylcholine is released from nerve cells of the parasympathetic nervous system. The endothelium responds to that acetylcholine, activates an enzyme that makes cyclic GMP. Cyclic GMP is a vasodilator. Cyclic GMP causes more blood to flow into the corpus cavernosum and the corpus cavernosum and the corpus spongiosum. Corpus cavernosum, corpus spongiosum. More blood flow, the blood, causes swelling, engorgement, erection. Cyclic GMP, vasodilator. Cyclic GMP is then, after a certain amount of time, is broken down by a phosphodiesterase. You've seen phosphodiesterases. We've seen phosphodiesterase that are sort of constitutively active. We've seen phosphodiesterase in the retina, right? Phosphodiesterase is necessary to break down cyclic GMP in the retina in order to cause the hyperpolarization of the retina. So you've seen this molecule before, but now it's got an interesting function. Well, interesting if you're interested in sex. It causes engorgement of the penis, erection, and it's broken down by a phosphodiesterase to reverse that process. It has, in the male reproductive physiology, there's a very specific isoform of phosphodiesterase. I'll bet you could guess what drug targets and inhibits this enzyme in order to prolong and increase the amount of cyclic GMP. You've heard it. It begins with a V.
Viagra.
There you are. Viagra works. I'm not going to test you on pharmacology. In fact, I'm not a... What do I want you to know from this story? I want you to know the general parts of the story. The brain, the autonomic nervous system, in particular the parasympathetic nervous system, cyclic G, that blood flow into these anatomical structures that I do want you to know the names of. So as long as I want you to know the names of them, I want you to know their function, and their function is erection. Viagra was developed, it was actually developed as an anti-baldness drug. They thought that stimulating blood flow in the scalp would actually help men grow hair back. It doesn't work that way. But they discovered a side effect. And they said, oh, we can make money on this side effect. And so they developed it as a drug for erectile dysfunction. It's a very funny detour that led to a lot of money for some people in a drug company. As you might predict, men are more interested in erectile dysfunction than they are in baldness, although, well, not so. Okay, erection and then ejaculation. Ejaculation is a response of the sympathetic nervous system. So stimulus, sympathetic nervous system, a nerve of the sympathetic nervous system. It's actually a mixed nerve. It's a motor neuron as well as a neuron of the sympathetic nervous system. I will not ask you the name of this nerve. Activates the bulbospongiosus muscle. Again, I asked you to know the name of the muscle. So now here's why. It's because a contraction, sort of a spastic contraction of the bulbospongiosus muscle, is responsible for ejaculation of semen. Semen is a mixture of sperm from the testes and non-sperm things, salts, sugars, fluids, from the prostate gland. That mixture is stored in the seminal vesicle, but then delivered through the prostate gland in response to this spastic contraction of the bulbospongiosus muscle. That's male reproductive anatomy and physiology and endocrinology. Any questions? There's a couple of clickers, which I'm going to not do for the interest of time, but you could do them as homework on Google. I didn't even print them out, so we're not going to go through them, but I recommend that you, in the process of studying this material for the final exam, use the clickers, and all your other tools.
Bob,
Yeah.
slide 16, it doesn't quite look like the bulbospongiosis contraction would have an effect on the vas deferens.
Yeah. So I'm not 100% sure this cartoon is fully accurate. I do think that there is muscle other than the bulbospongiosis muscle that also contracts, because you think that you need something to contract the seminal vesicle.
Right.
So there could be some myoepithelial cells here, myo meaning muscular, epithelium being epithelium. Myoepithelium, we're going to see that word again when we talk about the secretion of milk. There could be some myoepithelial cells that help the seminal vesicle not just be a bag of semen, but squirt some of that semen into the prostate. So the short answer is, yeah, you picked up on something that it doesn't look like this muscle here is going to help get this semen where it needs to be. And I think the answer is, yeah, you're right. There are some other muscles. There's some other contractile tissue that assist.
Gotcha. Thanks, Bob.
Any other questions? Okay, so some clickers. Let's take a 10-minute break. Move around a little bit, and then we'll turn our attention to female reproductive anatomy and physiology. Similar story, some commonalities, but some important differences, of course.